Evan and Benjamin, you guys are here. Yeah, I see you guys. I forgot to ask, which one of you would be sharing the slides?
I'll be sharing the slides.
Benjamin, so I have to turn you into a co-host. You are a co-host now.
Okay.
Excellent. Okay, everyone. Again, welcome to the last class of the year. Hope everyone is doing well. This is our last chance to participate, to actually help the presenters get better grades, because by asking them questions, it means that you really understood something. You're showing interest. You are indicating that you understood something, and it's going to look better for them because they're responding and they're challenging you. You are challenging them, then they're challenging you back. So that's all very, very good. So having said that, I'm going to pass on the mic to the presenters of the day.
Okay. Good morning, everyone. My name's Evan, and I'm here with Benjamin. And today we're going to be talking about a proposal, which is looking at the efficacy of viral nanoparticle-induced antimicrobial oligopeptides and combination therapies for the treatment of pathogenic oral biofilms. So today we're going to give a little background information on biofilms and the particular disease we're looking at. And after that, we'll stop with a question break. And honestly, if you have any questions within that, feel free to ask. And then we're going to get into our objective and hypothesis, and then our methodology and how we're going to achieve what we're looking to do, and then finish off with some possible obstacles. So going through with this presentation, we hope to at least relay information on you about biofilms and their importance in oral diseases. And then we'll look at some classical as well as some novel approaches to attacking the treatment of pathogenic biofilm. And then we'll look at viral therapy as well as antimicrobial peptides and how they can be useful within medicine. And then we'll give our proposed novel approach to treat oral caries, which is a form of tooth decay, using nanoparticles. So to start things off, does anyone know what biofilms really are? General explanation. Emily?
Can you guys hear me? Sorry,
Yeah.
Okay. Generally speaking, well, people usually hear biofilm at the dentist, so it refers to a thin layer of plaque or bacterial film on the teeth and gums, but it can really be made up of any microbial species if they kind of group together in a colony and form a film on something.
Right. So, does anyone else know? Okay, we'll move on. So yeah, basically that's actually it. That's getting more into our actual proposal. But for some general terms, we wanted to say that it's basically just a complex aggregate of microorganisms that can grow on many different surfaces, like Emily said. It's not just within the human body. They can be on things such as medical devices, and they can actually consist of one species of microorganism or multi-species colony that will cohere to each other while adhering to a surface. And they're actually held together by an extracellular matrix that is composed of extracellular polymeric substances, or EPSs. And again, these could be on many different surfaces and can actually be beneficial or negative. So biofilms may be seen in wastewater treatment, so that's a benefit of them, or they can actually harbor infectious agents in the human body and cause health issues or disease. So looking at the extracellular matrix and extracellular polymeric substances, or EPSs, they basically just influence spatial organization for cohesion of the microorganism. And they also include these specialized adhesives so they can attach to a surface. And they're composed of many molecules such as polysaccharides, proteins, extracellular DNA, and these could actually be biosynthesized by the bacteria itself or microorganism, or they can actually be sequestered from a host organism. And what they do is they actually act as shields They'll protect the bacterial colony from, say, antimicrobial agents, as well as a possible immune response from a host, or they can actually respond to microenvironmental changes from the host, such as pH change. And then here we just threw in a figure of different forms of biofilms. So, and it just adds to the complexity of biofilms. So even bacterial strains from a single species can have different morphologies according to certain environmental conditions. And because of this, the problem of treatment raises even more because
there is a question. So maybe one of you can keep an eye on the list of participants when someone raises their hand.
Sure.
Okay, yeah, good.
Yeah, I just have a question. Can any type of bacteria aggregate enough into a biofilm, or can only certain species and types of microbes develop into a biofilm?
I'm not exactly sure if every species of bacteria can aggregate into a biofilm. Maybe Benjamin knows.
I'm not sure if every single species of bacteria can, but like there's two different kinds of states that bacteria can be in. They can either be in a planktonic state, which is just free floating, and then they can be associated with a biofilm. Usually, it it's basically like they're forming colonies, right, that are forming together to like protect each other from the external environment. And through these EPSs that Evans mentioned, I'm sure that other bacteria can be inside of that biofilm, even if they don't themselves produce the extracellular matrix. So,
To my knowledge, it's not like all bacteria are going to do it.
right.
There are bacteria that don't. Now, like, maybe someone can claim that under specific conditions they may, but I don't think it's known.
Actually, just looking it up real quick, it is a universal trait of bacteria.
Oh, it says all of them they can?
Yeah.
Interesting.
Okay. So I guess we'll continue. So again, a single species can actually form different morphology of biofilms, and these different morphologies can actually make it harder to treat because, say, different particles and treatments will diffuse differently through the extracellular matrix, making it even harder. So looking at biofilm development, specifically oral biofilm development, it's generally characterized by 4 stages, where we have the initial adhesion of the planktonic bacteria, and this is actually dependent on the deposition of a salivary pellicle. And then moving into stage 2, this is when we get the cells beginning to divide, and they'll produce their EPSs, and they can actually recruit further planktonic bacteria to co-aggregate with the bacteria of the initial layer. And then in stage 3, the biofilm begins to mature, and this is where the EPSs actually develop into structural elements to protect them. And these could actually grow up to about 100 micrometers in length. And then when we've reached full maturity, bacteria are actually able to disperse in stage 4, and they can actually locate to other surfaces and start the process all over again. Hunter?
Just a quick question. So I was wondering about the physical robustness of these biofilms. So people like brush their teeth and stuff. If you brushed the biofilm, would it rupture, or is that not really the case?
I believe when like the kind of initial development of biofilm, so in toothpaste, there's sodium fluoride. These are things that actually can break up the biofilms and actually basically detach the biofilm from the teeth. But once we get into our sort of proposal, these biofilms can actually develop into hard substances like calculus on the teeth, and those are what's harder to detach. But
Okay, thank you.
okay, so moving on. We sort of mentioned it, but does anyone know sort of the importance and why we should study biofilms, maybe related to human diseases? Hunter?
Yeah, don't they have mechanisms related to antibacterial resistance?
Yeah, so that's kind of one of the reasons. So we go to the next slide, yeah. So kind of the significance of biofilms, like Hunter said, because they're usually said to be almost 100 times more resistant than the single bacteria themselves. This has led to increased antimicrobial resistance. And this poses a problem in regards to treatment associated with the biofilms itself. And factors that lead to resistance are the fusion of antimicrobial agents through the EPSs themselves. Also, bacteria can have enzyme-mediated resistance, as well as their overall structure of their membrane and efflux pumps to pump out antimicrobials. And these all contribute to the problem of resistance. And why they're so important in human disease is that they are estimated to account for about 75% of microbial infections. And some of these diseases include urinary tract infections, infections to wound sites, cystic fibrosis, and all forms of periodontitis, which are dental diseases. So looking at oral diseases specifically, again, biofilms are the main causal agent of diseases associated with in the oral cavity. Here on the right, we added a picture of a scanning electron micrograph of biofilm actually on a tooth surface. So that's pretty cool. And with oral diseases, it's actually estimated that 50% of the world's population will develop some form of dental disease at some point in their lifetime. And the economic burden attributed to this is immense. So in the USA alone, in 2019, this costed about 3.5 billion as a direct cause, with also an indirect economic burden of over $150 billion. So looking into a specific oral disease, we wanted to look at dental caries, which is basically just characterized as tooth decay caused by the pathogenic oral films within the oral cavity. And this is a chronic transmissible disease caused by the constant demineralization of tooth enamel. And this can produce tartar, like we can see on the right, which is the hard calcified substances that we can have on our teeth. And this overall produces decay in teeth, beginning with cavities usually. So it's the most common disease associated with the oral cavity, and it can affect at least 20% of children between the ages 2 and 4, and almost 80% of young adults. And another cool fact, it's not that nice, but 66% of adults aged 35 to 44 have lost at least one permanent tooth due to this specific disease, dental caries. So it is caused by bacteria, and the main causal agent is Streptococcus mutans, but there's also a lot of other species within the bacterial biofilms of the teeth that can contribute, but they're at lower concentrations like Streptococcus subrinus, Staphylococcus aureus, and various lactobacilli species. And what these associated bacteria all have in common is that they're gram-positive bacteria, so they have a thick peptidoglycan layer. They're also acid-producing bacteria, and they produce this acid through fermentation of sugar from dietary intake. And looking at Streptococcus mutans, they synthesize the majority of the biofilm EPSs, and they do this through encoding glucosyl transferase enzymes. And these biofilms not only affect the tooth surface, but they can actually affect lower into the root. Also, if someone has dental implants, and this can actually lead to further oral complications, such as chronic inflammation. So looking at current treatments, as we all know, antibiotics are failing for a lot of infections associated with bacteria, as well as biofilms. And sort of a new emerging field with treating oral biofilms is the use of nanomaterials or nanoparticles. So these are just particles that range from 1 to 100 nanometers in size. And there's kind of a scale there on the bottom. And then what these nanoparticles offer as an advantage, they actually offer administration under poor water-soluble conditions, as well as protect against enzymatic degradation. They can have greater targeted specificity to reduce collateral toxicity, and they generally have better permeability through biofilm matrix and biofilm membrane. So some examples include liposomes, metal particles, and more closely related to our proposal, bacteriophages and antimicrobial peptides. So what antimicrobial peptides are, they're small peptides that are actually in an organism, and they're important to their immune system. And they've been documented to have a wide range of inhibitory effects against bacteria, fungi, parasites, and also, for the sake of our proposal, biofilms. And they can either kill directly the bacteria, or they can actually modify a host defense mechanism, so the immune response. And these antimicrobial peptides have been reported to kill both gram-positive, so the bacteria we're looking at, but also gram-negative through mechanisms like destabilizing membranes and enhancing host immune function. And here on the right is just one form of how an antimicrobial peptide works. So this antimicrobial peptide in the video is actually inducing pores within a bacterial membrane, which is leading to nutrients diffusing out of the cytoplasm, and this actually kills the bacteria. So moving on, we wanted to talk about bacteriophages as well. So they're a form of virus that directly infects bacteria and can be about 24 to 200 nanometers in length. So they're generally characterized as a nanoparticle. And what they do is they actually penetrate and insert genetic material through a bacterium's membrane, and they can hijack the host transcriptional machinery, and through replication, they can lyse and kill the cell which they infected. And this infection of the bacteria, the replication occurs at a very fast rate, and these replicated phages can actually attack the surrounding bacteria as well. So that would be good for biofilms. So another important factor that goes with our proposal is that bacteriophages are actually able to produce depolymerizing enzymes like endolysins that can also degrade biofilm EPSs. So that's basically all our background information we wanted to say about biofilms and everything else. So we looked at the impact of biofilms and how they're apparent in medicine today, and then also the development and treatments that are around today, and then sort of getting into our proposal, looking at bacteriophages and antimicrobial peptides and how they can possibly be used to combat biofilms. So again, that's basically all our background information. Does anyone have any questions before we move on?
All right. We'll get into our
Oh,
actual.
Mandy has a question. Sorry.
In terms of matrices, do they only grow on flat surfaces? Not flat, but like, do they only grow in like solid surfaces, or can they also grow in like, let's say, surface waters or anything that's not like constant?
I believe they can grow on not just solid surfaces, but I'm not 100% sure.
Yeah, well, if we go back to the slide that is talking about the stages of biofilms, you can see that there's a part where they actually disperse and they're fully encapsulated by the extracellular matrix. So I could see a potential scenario where the bacteria are growing within that dispersal, or like not on a solid surface. And yeah, so as long as it's a co-aggregate of bacteria, they can form that extracellular matrix and form like a biofilm, basically.
Jaden?
I was wondering, I think it's really interesting how that nanoparticle kills the bacteria. Do you know how prevalent that is, like in use right now? Is it used a lot?
I'm not sure for all types of biofilm development, but when we were looking into the oral aspect of it, it's like generally within the past like 2 to 3 years, that's been the new sort of emerging field to combat oral biofilms. I'm sure it'd be the same for other biofilms as well. But yeah.
Mandy?
Yeah, just to add to like Jaden's question, um, there are there's a lot of like nanoparticles that are like being studied to like in terms of antimicrobial application. A
Mhm.
lot of them are actually like being used into like a lot of like products that usually um needs for like preservations and stuff like that. One of
Right.
them is copper oxide.
Yeah. Another part, or we're going to mention in our thing, they've been used for food preservation as well. So
Emily?
So what mediates the transition from like a biofilm or plaque to a calculus? Is it exposure to oxygen rich environments, or is it the calcification of teeth?
yeah.
Go ahead,
Oh,
Emily.
sorry. Go, Ben.
So the calculus is formed from the remineralization of, or the like recrystallization of the minerals that are in your tooth enamel. So if we go back to the slide, you can see that like this is really crystalline, right? And so this is just like the minerals. This isn't necessarily just made out of the proteins and extracellular DNA and such that make up the biofilms. The tartar and plaque that we were talking about is mostly formed from the recalcification of minerals that were dissolved by the lactic acid that the bacteria produce through um, metabolizing the sugars that they eat. Syed?
So I just got a quick question. You said we'll be using nanoparticles to treat the bacteria. So my question was, I recently I was reading some articles in which they says that nanoparticles are causing cytotoxicity as well when it comes to treating something in the body. So what do you think in this case, how they will be impacting this cell?
Yeah, well, there's a bunch of different types of nanoparticles, right? So it just depends on like how large it is for it to be considered a nanoparticle. Like we mentioned, there's heavy metal elements that you can use. There's viral agents. There's um, antimicrobial peptides that are small enough to be considered nanoparticles. Um, and yeah, so there's lots of different ways in which these nanoparticles can act. Like the antimicrobial peptides, for example, they can have there's certain ones that are alpha helices that can break down the cellular membrane and cause poration. Um, I'm not exactly sure what the mechanism for the heavy metal elements are. Um, but yeah, we'll get more into how exactly the our antimicrobial peptide breaks down the bacteria. Does that answer your question, or did I kind of miss the mark on that?
A bit, but I was wondering how they're gonna like the nanoparticles that you guys are gonna use will be impacting the cell. Like what will be the effect on the cellular level?
Oh, okay. We'll get into that once we actually describe what
Right.
nanoparticle we're using, or the
Yeah.
antimicrobial peptide rather. Are we all
Okay.
set? Okay,
Yeah, I think so.
cool. All right, so moving on to antimicrobial peptides and bacteriophages, just a quick overview again. Um, so both of these have exhibited antimicrobial effects and can be used as an alternative to antibiotics. As we've mentioned, it's an emerging field. Well, the nanoparticles specifically are, just because of the new strains of antibiotic resistant bacteria. The great thing about bacteriophages is that you can genetically engineer them. And the basis that our proposal is based on is that we can actually insert a cassette which encodes for the antimicrobial peptide. So we're going to be throwing the genetic code for the peptide inside of the virus. So when we infect the cells with the virus, we're going to be having the bacteria produce the antimicrobial peptide, which will go on to ultimately kill the cell. And so the bacteria will reproduce more viruses, which in turn will produce more antibacterial peptides, which will have an overall cascading effect. As shown here, there's actually another study which we did not include in the reading as we found it just after we submitted our proposal, but there was a T7 bacteriophage encoding for what is known as the 1018 antimicrobial peptide, and it was shown to increase the efficacy against both planktonic associated biofilm E. coli. In our approach, we're planning on using an antimicrobial peptide known as nisin. It's first in the form of nisin A, which is a non-bioreactive form of nisin. It needs to be post-translationally modified for it to become actually bioactive, but we'll get into that later. And the bacteriophage that we're planning on using is denoted as SMHBZ8, and yeah, move on to actual bacteriophage. So bacteriophage SMHBZ8 is one of only 4 described bacteriophages for the bacteria S mutans. We have M102, M102AD, which is just a mutated form of M102, OAPCM01, and the most recently found SMHBZ8. It was first described in 2021, so last year, by a team in Israel. And the literature shows that it's more robust and has greater lytic capabilities than the other 3. All of these bacteriophages belong to the family Cephalophoridae with a diameter of about 60 nanometers. This makes it a good choice for attacking the S mutants associated biofilm associated in the oral cavity. On the right, we can see just a diagram and a electron micrograph of what is known as a lambda phage. On the top, the bar represents 100 nanometers, and figure B on the bottom there is actually a TEM image of the bacteriophage that we're planning on using. As you can see, they have similar morphologies with the same capsid and tail. Yeah. Just relating back to how it is a nanoparticle. Here we can actually just show you a little bit of the antibacterial actions of our bacteriophage. These are SEM images of S mutant biofilms after 24 hours and 48 hours after the virus was added. And you can see that there's a significant decrease in the amount of bacteria and the associated biofilms. Moving on to our antimicrobial peptide. It's an antibiotic, which is just a polycyclic peptide antibiotic. It's an antimicrobial peptide which is used extensively in the preservation of food, as well as other applications. It inhibits the growth and kills multiple gram-positive bacteria, including S mutants and others that are found within the oral microbiome. The other effects that it can have are anti-gram positive, which is what S mutants is. It also has spermicidal effects and can work synergistically with other AMPs. It also works against the biofilms. It's found to have wound healing capabilities, and it's also an anti-cancer agent. So the way that it works is it creates pores within the membrane of bacteria, when it's in its bioreactive form. And what this does is it just makes it so that the bacteria isn't able to form ionic uh, kind of gradients. So it just makes it so that the cell sort of falls apart. The efficacy of nisin is increased with sodium fluoride. This is pretty good for us because sodium fluoride is already an agent used extensively within dentistry. I'm sure tons of your toothpaste and mouthwashes already have it in it, so it's already an agent that we're using regardless. It's odorless and it's tasteless, which is great if we end up using this in a in vivo setting with like an actual application, as it wouldn't leave a an off-putting taste in your mouth. And for these reasons, successfully genetically engineering a phage encoding for nisin in combination with sodium fluoride will hopefully increase the efficacy of treatment against biofilms associated in the oral cavity. So the general objective of our proposal is to determine if the combination of the endolytic action of our phage in the with the active form of nisin will be more effective at eradicating and preventing the formation of mono and multi-species pathogenic oral biofilms than any of one of these therapies alone. As mentioned here, we're also going to be looking at how sodium fluoride has possible synergistic effects, and we're hoping to see close to 100% eradication of the biofilms. As you saw on that previous slide, the phage alone is pretty good at getting rid of these. Obviously, in a practical setting, in an in vivo setting, this might be a little bit different just because you won't have a very, very concentrated attack on them, whereas you would have in an in vivo setting. But that's for a later discussion. Our hypothesis is that the efficacy of eradicating the biofilms is going to be increased using this combination approach. And this is backed up by current literature. There's been studies to show that the combination of different antimicrobial agents has had a synergistic effect. And this is just one of the papers that have shown that you can encode for a antimicrobial peptide within a bacteriophage. So getting into the methodology of how we're actually going to go through with our experiments. Firstly, we're going to be looking at bacteria and phage acquisition and the proliferation of those agents. So the bacteria that we're using, as we've mentioned, is S mutans, and we can just order this from suppliers. As for the multi-species culture, we're going to be planning to use samples taken from volunteers, and various other media can be used for proliferation, and we're going to be getting into that in the next slide. The phages can be acquired via collaboration with the institute that first described the virus. Though if that doesn't come through, then what we can do is something called rebooting the phage, where we essentially synthesize the phage from scratch. Proliferation done in growth media containing live S mutant cells for this phage, and they'll be separated via centrifugation. I actually emailed the institute or the lab team that first described the phage. They haven't gotten back to me, but I would assume that they most likely have some of these phages either frozen or they've archived them in yeast genomes. So we'd be able to, if they did decide that they wanted to collaborate with us, then that would be a great leg up. So the bacterial proliferation media that we're going to be using is brain heart infusion broth. It's worth noting that there are multiple steps that would be included in the actual study to determine which strains of bacteria were actually present in the multi-species cultures that we're going to be getting samples for. But for the sake of time, we're just going to omit those details. As far as acquiring the viable phages, as we said, the collaboration between laboratories is a possibility and a preferred route, but we can go ahead and reboot the phages if necessary, which we will explain later. For the phage proliferation, it's going to be done in the same media, like we mentioned, though this new media is going to be containing the S mutant cells. This is just because the bacteriophage is an obligate phage of S mutants, and we need that bacteria to actually grow the viruses. Onto the phage genetic engineering methods. We have both in vivo and in vitro options. Both are legitimate ways, but there's some things to consider for both. Firstly, getting into the actual cassette that we are introducing into the phage. Within this genetic cassette, we are going to be encoding for GFP and nisin. This is integral to our study as nisin is the antimicrobial agent, and GFP is going to be used to both determine if the cassette was successfully inserted into the phage genome and to identify bacteria that are expressing the genes on the viral cassette. Later, we'll discuss that we're actually going to be using a Western blot to see if the bacteria are actually producing the nisin, but making the antibiotics that are specific to nisin and making the gel is an intensive process, and it's much easier to just quickly check before we do that for fluorescence through checking for GFP. Getting into the recombinant technologies that we would be using to actually get the cassette into the virus, we would have a plasmid containing the cassette. Also have homologous segments flanking either side of the actual cassette. That would be homologous to parts on the viral genome. So using this, we would just use homologous recombination, the same as you see in meiosis, for example, to get the viral cassette or the cassette into the viral genome. I'm not exactly sure, but I believe that there's an enzyme that would be needed for this recombination to occur, but we'll need to look further into that. Because the virus SMHBZA belongs to the family Cyphophoridea, it's reasonable to assume that the phage has a capsid capacity of around 50 kilobases, as this is the average for the family. It's also therefore reasonable to expect that a double-stranded DNA with the additional codon optimized genes for the expression within S mutants encoding for GFP and pre-nisin, which is nisin A, would also fit within the capsid, as the wild type DS DNA strand contains only 32 kilobases. The transgenes would only account for an additional 1 to 2 kilobases, depending on the operons that we use, so this is negligible. As far as rebooting the phage goes, this is a technique where you actually synthesize the phage from scratch. So there's a couple of ways that we can do this. We can do this in vitro by ligating individual genome fragments cut with specific restriction enzymes. So we would just get a lab to produce the different fragments. But as you can see here with the transcriptome of the phage superimposed over the genome, there isn't a whole lot of space for us to cut and segment. And with ligation, there's always a chance that there's going to be mutations. And down here, we can just see the total number of restriction digest enzymes that cut only once on the genome. So we'll have to have 15 different enzymes, and this can just get messy. So we decided to go for a in vitro approach. where the full-length genomic DNA can be assembled with overlapping genome fragments using transformation-associated recombination, or also known as TAR, which exploits a high level of homologous recombination within yeast cells. The overlapping viral fragments can be amplified by PCR, with each adjacent fragment having a homologous sequence overhang. The first and last fragments would contain homology to the yeast vector that we decide to use, just for easy ligation into the vector. When transformed into the yeast cells, the fragments can be recombined to form a complete phage genome in the yeast vector. And this DNA would then be extracted from the yeast and transformed into bacterial cells to generate the phages. The great part of using this technique is that mutations can be introduced into any part of the fragment that we desire. So we can throw it into a intronic part of the genome and not really affect any of the functional genes. This would also, as I just mentioned, it would allow us to insert the cassette, allowing us to have the bacteria biosynthesize the GFP, nisin A, and the enzymes needed to post-translationally modify nisin A into the active forms of the peptide. And if you guys are interested, there are 5 different forms, or 5 different enzymes rather, that do this modification, known as NIS-Z, NIS-B, NIS-C, NIS-T, and NIS-P. The DNA extracted from the yeast would need to be transformed into L-form bacteria, as the bacteria that we are using is gram-positive. L-forms are just bacteria that have their peptidoglycan cell wall removed, so this allows for larger pieces of DNA to be readily absorbed. As you can see on the right, this is just a quick diagram showing the different forms of L-form bacteria with partially segmented cell walls and just fully completely gone cell walls. And these other 2 diagrams are just showing the different types of recombination that we can do, and also the this is the rebooting of a phage. So the introduction of the DNA into the L-form bacteria, which would then produce all of the peptides that are encoded on this DNA, and thereby producing the recombined phages. On to how we're going to actually run our experiments and collect the data. We're going to be going through the biofilm growth media and the quantification of our results. It's important that we try to mimic the oral microenvironment so the data collected is actually applicable to the real world. And for this data to be applicable, we must also have accurate and thorough ways of describing our results. The biofilm growth surface and media that we're going to be using, as we previously discussed, is going to be a tooth surface analog. And that's going to take the form as a collagen-coated hydroxyapatite disc. It's just a disc that mimics the surface of the tooth enamel. There are many types of media in which the biofilms can be grown, but we'll be using a combination of different media. The combination media will include saliva that we can collect from volunteers, and we can also somewhat supplement the volume of the media with additional different medias that have growth factors within them. For oral biofilms to develop, the dental pellicle, which is mostly composed of salivary proteins, needs to be deposited on the tooth surfaces. And because the artificial media lack these proteins, it's ideal to use saliva, which is why we're actually going to be collecting them from volunteers, pasteurizing them, and subsequently supplementing the volumes with additional media. As far as, oh, I should also go through our actual figure here which shows the what these discs in question look like. For the qualitative data collection, we're going to be using scanning electron microscopy to actually look at the development and degradation of the biofilms. On the right here, we can see an example of what one of these SEMs would look like. We would also be using GFP to see how much of the bacteria are actually infected with the transformed non-wild type viral vectors. We would also be doing live dead viability staining, followed by confocal scanning laser microscopy, which can be seen down here. This is an example with the red cells indicating dead cells. So we can see here that there's, this is just an example from one of those previous studies of the SMHBZ8, the viral vector that we are using against some bacteria. As for our quantitative data, we'll be using a Western blot to identify the amounts of the nisin proteins actually expressed. And we'll be using flow cytometry to be as an additional step to actually quantify the number of viable cells that remain after before and after the treatment of our combination therapies. Onto some potential obstacles that can arise during our experiment, some of which we have already addressed through the experimental design, and some that we still kind of have up to question. As far as our antimicrobial peptide goes, there's always the possibility that there's proteolytic enzymes or different salivary components that may actually degrade the antimicrobial peptides. And in an attempt to mitigate these effects, we've chosen to use phages because they allow for very localized and concentrated specific delivery of our antimicrobial peptide. So actually within the biofilm is where it's going to be expressed and released by the bacteria, whether that be through the lysing of the bacteria or through the bacteria's efflux pumps. And because they're going to be so localized, they won't be encountering a lot of salivary components or proteolytic components that may degrade the AMPs before they actually reach their targets. As far as bacterial resistance goes, as we all know, with the emerging technology that is CRISPR, bacteria have immunity, and with this they can adapt and gain immunity against bacteriophages. So there's a possibility that the bacteriophage that we are using could at some point become not viable just because the bacteria would become sort of immune to it. That's not most likely the case, as the virus that we are using is a relatively new species. Bacteria can also resist the phage attack through spontaneous mutations, restriction modification systems, and as well as just being in a biofilm makes them more resistant to the bacteriophages. As mentioned before, the biofilms are up to 100 times more resistant than their planktonic forms. That being said, phages are small entities and they can penetrate into the pores of the biofilms. So the biofilms shouldn't pose too much of a inhibitory effect on the efficacy of our approach, just because they are nanoparticles and they can actually penetrate into the biofilm. As far as the eradication and reinfection goes, there's the possibility that the bacteria won't be fully eradicated. That's kind of our expectation in an in vivo setting. And because of this, there's always the chance of reinfection, and then those reinfected, or the bacteria rather, that would reinfect the host would most likely be resistant to the approaches that we've already tried to use to um, eradicate them. The results of our trials remain to be determined, and we may adjust according to the data acquired. So the antimicrobial peptide that we decided to use isn't necessarily going to be the most effective against all of the multi-species biofilms, and it's just going to have to be a process of trial and error with that. Um, yeah, so there's also possible side effects as far as this therapy goes, as the bacteria that we are planning on eradicating from the oral microenvironment aren't just available in your oral microenvironment. They're also on your skin. Some are within your GI tract. So there's the possibility that this could have negative side effects on people who actually go through this treatment, or other macroorganisms that go through this treatment. But that's a problem that we're going to assess later in vivo trials. Right now, we're just focusing on the in vitro setting. Another possible obstacle is that there may be complications when it comes to biosynthesizing active niacin within the bacteria. If this becomes too large of a hurdle to overcome, we'll be transitioning into using pH-sensitive nanoparticle carriers, also known as NPCs, with a high affinity to exopolysaccharides, excuse me, as the effectiveness of this combination therapy can still be studied, even if the actual antimicrobial peptide can't be biosynthesized in situ. We were planning on using a different antimicrobial peptide before the one that we found. And this is where this obstacle actually comes in. Some antimicrobial peptides aren't able to be produced within bacterial cells just because there are multiple factors that go into creating a peptide that is actually bioreactive. Things that can go wrong are like incorrect folding of the peptide. There could be different actual proteins that you need to post-translationally modify or actually synthesize the um, peptides that aren't available within bacteria. Specifically, there is one known as AP00166. It was one found in a flounder fish, the mucosal skin excretions of fish. And we weren't able to use that just because it wouldn't have been able to be produced within the bacteria. So something to consider. And that completes our presentation. Thank you so much for your time, and we have some time for some final questions. Emily?
Excellent. Thank you very much. Please join me in thanking them for their excellent presentation. Any questions, comments, issues?
Isabel.
I'm sorry, I don't know if you guys mentioned this, but I was just wondering, do you know how quickly these bacteriophages could target the biofilm? Because I was wondering, like, if we somehow like find a way to apply it directly to the like teeth or mouth area, like how would we get it to like stay there long enough? Because like could people just end up like eating it up or
Right.
rinsing it out somehow?
Right. So what we were thinking is there could be an instance where it could just be like an oral rinse. As long as the bacteriophage actually penetrates some of the bacteria, that bacteria is going to be infected with the bacteriophage. And as soon as that happens, the bacteria is going to be producing more of the viral agents, and it's going to be producing the um, antimicrobial peptide, which uh, it will continue to do so until the antimicrobial peptide gets to a concentration high enough that the um, cell lyses, or due to the endolysins that the bacteriophage produces, it will burst. Um, but you should see immediate infection as far as infecting the bacteria goes. And once it's in the biofilm, it's just a cascading effect. It keeps on proliferating and infecting other cells that are within the same biofilm. Um, so yeah, it should be rather quick. I don't know exactly how long the rinse would have to be or how long you would have to keep it in your mouth, but that's of course um, something that we could look in our in vivo and in vitro settings. We could introduce the phage for um, just maybe a minute or so in our in vitro setting, and then wash away the media and replace it with new media and see what the effects against the biofilms are. So, Jin?
You said in the possible obstacles section that the biofilms are actually 100 times more antimicrobial resistant than regular planktonic, which I found pretty interesting. So
Mhm.
I just want to know if you'd be able to explain kind of why or some detail about it.
I actually wasn't the one who did the research into that. Do you know?
Yeah. Um, I think it's basically just to do with the extracellular matrix. So when you have, say, gram positive or gram negative bacteria, the penetration problem for antimicrobials is usually due to either the peptidoglycan layer, but in biofilms, they have these buildup of the extracellular matrix that have these really tiny pores that make certain antimicrobials very hard to penetrate within. And that's way different than just say, a single bacteria itself. So kind of that, if that answers your
Yeah,
question.
there's also like there's tons of different antimicrobial agents. Some are rather large, and that can just inhibit the effect that the biofilm has, or the antimicrobial um, peptide or agent has against the biofilm, just because like it's covered by the extracellular matrix, right? So it has less of a chance to actually penetrate the bacteria. So whereas in the planktonic cells, it doesn't have that extracellular matrix, right? So it's fully exposed and around um, all sides.
Right.
No, Noah?
You said this would be used in conjunction with fluoride and at the dentist or something. So would this kind of treatment only be done at a dentist?
No. So what I was saying with the sodium fluoride actually is you can already just buy fluoride and have it as an oral wash. Like you can,
Yeah.
right? So what we'd be planning on doing is using the sodium fluoride in conjunction with the viruses. So it would
Mhm.
just be like a cocktail of just like a mouth rinse, and then hopefully the sodium fluoride has the synergistic effect that's expected with the antimicrobial peptide.
Well, so I know research would be done, so there'd be no drawbacks if people accidentally drank it, or like would it affect their healthy bacteria in their stomach?
Yeah, so that was one of the potential obstacles that we were talking about, the side effects.
Yeah.
We're not too sure exactly what the total effects would be. So the uh, the bacteriophage in question is very specific, right? And it only infects the the one bacteria. So you don't have to worry about it having a cascading effect against like all of your beneficial bacteria.
Yeah.
Um, and so as soon as the populations of the S mutants is degraded, the amount of antimicrobial peptide is also going to be diminished. It should also be noted that the antimicrobial peptide is rather safe. It's currently used in food preservation, so like you've probably already eaten it. Um, so yeah.
Perfect, thanks.
Excellent. Thank you very much. Please join me again in thanking the presenters for their presentation. And at this point, we're gonna take a break. Ben and Evan, I'll meet you guys in our meeting room. And everyone else, see you guys back in about